Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Luca Giorgetti from the Friedrich Miescher Institute on this show. Please let me briefly introduce you to our audience. You are physicists by training and you obtained your PhD with Gioacchino Natoli at the European Institute of Oncology in Milan, where you combined physical modeling with quantitative experiments to characterize the inflammatory transcriptional response. You then did your postdoc with Edith Hurd's lab at the Curie Institute in Paris. And then in 2015, you started your own lab at the Friedrich Miescher Institute for Biomedical Research in Basel. And you are still there today. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
I think I got involved. I got interested in biology quite late compared to other people I know, because I studied physics in the beginning, and then I did a little bit of short research experience in theoretical physics, so absolutely nothing to do with biology. But then, for different reasons, I landed in a lab at the University of Milan, where they were characterizing nanostructure surfaces for biology, for biocompatible materials. So that's how eventually I got in touch for the first time, really, actually, with cell biology and molecular biology, because we were collaborating with a team at the European Institute of Oncology in Milan. And I was working more on the material science side of these surfaces. And they were mostly culturing the cells and looking at what cells did when they grew on the surfaces. But then I got really excited about the biology part itself, and that's how I actually eventually got a PhD position at the European Institute of Oncology. So working sort of at the interface between material sciences and molecular biology. And this was in the group of Paolo Milani back then. But then while doing this, I actually bumped into Gioacchino Natoli, who was still a group leader at the European Institute of Oncology. Who actually was working on, so he's world leading expert in transcriptional control and inflammation and immune system. And so, and he was actually looking for somebody who might help on project that they had in the lab that got a little stuck. So they had a collaboration with Marta Bulik's lab in Harvard Medical School and Manolis Pasparakis in Cologne back then. And the idea was that they had some, so they found that in NF-kappa B target genes, there were clusters of NF-kappa B binding sites that nobody knew what they were used for. They didn't know actually what to do with this information. So Gioacchino proposed that I actually try to do some modeling for them. And that's how I started drawing some cartoon models for transcription factor binding and how it affects transcription. And then somebody actually had to test these models, and that's how I began doing some experiments as well in the lab, and that's how it all started. Yes.
So let's come.
Random walk, I would say.
Serendipitous, or how you call it.
Indeed. Yes.
Okay, so coming to a sign that centers around, very generally speaking, chromosome structure and transcriptional regulation. But let's start in the year 2014, because this is when I think thematically the work that you are currently focusing on started. So to paint the picture, this was the time when TADs were the new kid on the block and were discovered using 3C. You worked with Edith Hurd and set out to explore TAD structure and developed a polymer model that can extract the full repertoire of chromatin conformations within TADs from population-based 3C data. Can you maybe talk about this work, maybe also transitioning from what you shared just now and what you found?
Yeah, so yes, I got into chromosome structure and transcriptional regulation coming from Gioacchino's lab, because during my PhD I actually worked on transcriptional regulation by NF-kappa B. So, and then I became interested. So when I looked out for postdoc positions, I already knew I would like to work more on chromosome structure and transcriptional regulation, because of course even back then, even before TADs were known, we knew that distal enhancers target genes that are very far away in the genome. And of course, there was a lot of evidence already that physical interactions actually matter in this process. And then, so I then joined Edith's lab at the Curie in Paris. Right when, I would say, Alfej Nora, who was a student in Edith's lab and now is his own lab at UCSF, actually discovered the existence of TADs. So it was an incredibly exciting time. So I have to say, even maybe before I knew about TADs, my idea had always been to sort of use polymer models to deconvolve single cell trajectories of the chromatin fiber out of any type of 3C data. So that was what I really wanted to do in Edith's lab. But then, of course, when Alfej discovered TADs, it was such an amazing discovery and such an important moment that sort of the stars converged once more, and I could bring in my tools to actually study chromosome conformation and transcriptional regulation in a very relevant system, which is actually back then was the X chromosome, so the X inactivation center the locus on chromosome X that controls essentially the onset and maintenance of transcription of exist, which is the master regulation of X chromosome activation. And of course, Alfej discovered that there are two TADs that control that contain essentially the regulatory elements of exist on one hand and of TSIX, so the antisense RNA that controls there is a negative regulator of exist on the other hand. And yeah, so that's how I ended up using polymer models in the first place.
Yeah, what does it exactly mean, like the polymer model? How does it describe the TADs or the chromatin structure?
Well, I'll say, nowadays we know much more about the mechanisms that give rise to topological boundaries in mammalian chromosomes, and we know about loop extrusion and how it's blocked by CTCF sites and how this results in asymmetric contact preferences across genomes. But back then, the first sort of evidence for TADs was based, in Alfej's case, was 5C based. And in Bing Ren's lab, and also Giacomo Cavallo in Drosophila showed that the same similar structures as well in human cells and in Drosophila. But of course, the resolution of these experiments were relatively low. So we had a resolution maybe of 10 kilobases or so, a little bit more even. And we weren't able to see CTCF interactions directly. This came later, actually. And so one of the questions that immediately the discovery of TADs brought up was, what are they? What do they arise from? So what molecular mechanisms actually could give rise to asymmetries in how sequences interact in the genome? And the other question, of course, was what do they really represent in single cells? Because what 3C methods do generally is that they average over millions of cells, at least in their population average versions. And so of course, one could use single cell experiments, for example, DNA fish at high resolution, to look into the structure of the chromatin fiber within single domains, which is actually what we ended up doing. But in the beginning, the idea was really to try to use computational models to address both of these questions. So what do TADs arise from and could arise from, and what they actually represent in single cells? Because one of the ideas back then was that, of course, these are sort of blobs that are relatively identical in all cells and host pre-formed contact between promoters and enhancers to some extent that are pretty much identical in all cells. And so the idea there was to try to use polymer. So of course, chromosomes are polymers, very long polymers. The chromatin fiber is a polymer, and so the idea had been to really try to use simulations of polymers and compare them to what actual 3C experiments show, in order to find potential mechanisms that you can show on the toy polymer give rise to the same features, and hence identify potential mechanisms that give rise to the structures that you see in 3C. And on top of that, these as a goodie, this would actually have the advantage that these simulations describe the actual thermodynamic ensemble of conformations that give rise to those average contacts that we observed in 3C, and hence we could use them to deconvolve the single molecule conformations.
So this was at first the physicist in you that tried to understand how TADs were formed. Could it be described like that?
Yes. So I should also mention this was done in a collaboration with my good friend and longstanding collaborator Guido Tiana at the University of Milan, who is actually a biophysicist. Now having said this, yes, it was my idea in the beginning to actually use the simulations to get at potential mechanisms. But of course, we didn't get at the actual mechanisms. This only came later. What these simulations showed, I think, is that two important pieces of evidence. So the first was that you can create, so that these blobs or whatever regions of preferential interactions could be created just by sort of attractive interactions within each domain, so that you didn't necessarily need something at the boundary that separated them, that pushed them away actively in order to create them. So internal interactions were just enough to make them happen. And the second thing that they showed is that there was a substantial cell-to-cell variability in the structure of the chromatin fiber within each individual TAD. So contrary to the maybe naive view that each TAD was actually a little bit of a blob. So our simulations showed, and then we could prove using high-resolution DNA fish at the XIC that this was indeed the case, that there is very large amounts of structural differences between individual cells where the experiment is performed. So, which had important implications, I think, because it showed, at least formally, that it's not like in every cell, every enhancer and every promoter are in contact all the time and in an identical manner, but rather that enhancer promoter interactions that occur within TADs are also stochastic events that are only occurring at any given time point in an individual subset of cells. So that was what we could definitely show. Yes. And now, of course, we know about the interactions that established TADs much more, because it's a very passionate story as well. Somebody else should tell you probably, because it's not what I discovered, but rather comes from Leonid Mirny's lab, and then based on Kim Naismith's and John Marcus as well. Yes. And it's looked by cohesion. Yes.
So you then started your own lab, and you looked into the regulatory microenvironment of TADs. You developed a new algorithm named CATCH. Maybe this also ties into what you just shared with us, that identifies hierarchical trees of chromosomal domains in Hi-C maps. So how does this work, and what did you find using this new algorithm?
Yeah, so the early days of my lab were still in the phase where we knew and didn't know too much about CTCF and loop extrusion and how TADs are established, but and oftentimes we were sort of debating. So the field was actually debating on what is really a TAD and what is not a TAD, because even with relatively low resolution Hi-C data sets, it's possible to see that it was possible to see that specifically highly active TADs. So located in regions of the genome where there are several active enhancers and promoters and CTCF sites, there were sort of substantial numbers of substructures that one could start perceiving. And so one of the things we did with my student, with my first student actually, Zan, was to try to maybe overcome this limitation of only looking at one of these layers and claiming that those are TADs and the rest doesn't matter, but rather to actually look at Hi-C maps with a more general approach in which you just scan through the hierarchy of the structures and try. And the question we asked. So first of all, we wrote an algorithm that allowed us to call domains at all of these levels, not just at one scale. And we asked the question using these domains called at various resolutions, whether there was any of them that had some more interesting properties from the biological point of view. And this was actually quite interesting because we saw that indeed the scale at which what we would have naively called TADs, so regions of the genome that are around 400, 600 kilobases in size on average in mouse genome, have maximized several interesting properties. So they're maximally enriched in CTCF clusters at their borders, show higher co-regulation of gene expression during development, And they're even more conserved throughout cell type. So it was an interesting exercise to show that despite the fact that there is structure at all levels that in mammalian genomes, indeed there's one scale or a few scales that emerge as being maybe more interesting biologically, and this scale is the one that is demarcated by strong clusters of CTCF sites, which is what I would now call maybe topologically associative domain.
Okay.
Yeah.
So next you worked on an alternative method to 3C called DAMC. And this is a method based on DAMID, which was developed in Bas van Steensel's lab. Why was it necessary or why did you want to have an alternative method to look at the same thing? And what did you find then using it?
Yeah, so this is also an interesting story because, of course, another of the questions that were highly debated at the time I started in my lab was whether really TADs exist or aren't just a cross-linking or ligation-based artifact, because of the way 3C methods work. Might introduce substantial biases in the kind of physical interaction that we detect. And so, especially when I started my lab at the FMI, Susan Gasser, who was director back then, so she kept asking me this question because we're very interesting. So, how sure you are this is not cross-linking artifacts? So obviously, these also motivated us to try to think about other methods. And indeed, when I was a postdoc in Edith's lab, when I was finishing, there were already some discussions with other lab members about trying to use DAMID as an alternative method to look at chromosome interactions. But then sort of we dropped it because this is not really feasible. But then eventually, so one of my first students, actually Josef Rodolfi, was eager to give it a try. And indeed, what we did was that we recruited down, so the bacterial adenine methyltransferase, to specific regions in the genome by fusing it to TET repressor and inserting at random places TET repressor, small TET operator arrays. And by essentially comparing the signal you get when other chromosomal regions get in touch with these recruitment sites of DAM with some background methylation levels, we were able to extract profiles, so contact profiles that essentially resemble to some extent 3C profiles, except that they are based on DNA methylation instead of being based on cross-linking and ligation, which has the advantage that the signal generation happens in living cells, and B, that it doesn't require any really biochemical procedure to detect the contacts except digesting DNA with an enzyme, methylation sensitive enzyme, so which reduces the number of potential biases. Of course, there are biases in that too, and we were lucky enough to be in touch with both Bas van Steensel, who actually provided a lot of fundamental insight in actually how to do these experiments and encouraged us to pursue them, and also with Wouter de Laat's lab, who were also doing something very similar. So we ended up collaborating on this, and we were able to, thanks to Wouter, actually to compare these profiles based on methylation readouts with 4C profiles. And to our bigger surprise, I mean, we were expecting that they completely differed because indeed of potential cross-linking artifacts, but in fact, they were very, very similar. And we were really surprised to see that and very reassured at the same time, because it meant that to some extent, but to a large extent, actually, cross-linking based 3C measurements can be trusted as measurements of contact probabilities across mammalian chromosomes. So I think it was nice to have done this and showed this formally, at least. And so this was really also instrumental for what we did later. Yes.
Yeah, it's really nice to see, because like in the episode that will have been, I must have been careful how to phrase this, will have been published 2 episodes before yours. I was talking to Alistair Brittinger, and he like looked at TADs with microscopy approaches, and now you are doing like not the same, but a similar approach using another method, just investigating whether TADs are real and how those things or how those methods show the same thing. And it's really nice to see that like 3, I wouldn't say like orthogonal methods, but at least different methods really show the same thing, which is nice to see, right?
Yes, and I should also mention that in Alferge's original paper, so he also performed quite a lot of high-resolution microscopy to actually look at whether really at the TAD boundaries that he identified at the XIC, he could detect physical insulation. It was already quite clear that these things were really third, that this separation, of course, occurred in single cells. But of course, I mean, now with the ORCA and other chromosome tracing experiments, it's become fundamental to prove that these patterns really do exist, and they are essentially probabilistic in nature. Yes.
Mm-hmm. So another question that arises is whether those chromosome interactions are actually translated into a transcriptional output in some way, and whether transcription is somehow affected by how the chromatin is formed. So what was your approach you took to investigate this, and what did you find then in the end?
Yes, so here I think I have to say, so when I started my lab, we really wanted to go at maybe more functional experiments that could prove whether yes or no chromosome interactions are causal to transcriptional events. Of course, maybe a little bit naively, but it was a sort of a. I think so. When I joined FMI, of course, is a great place to do sort of high risk, high gain projects, and we thought we should really push a little bit the boundaries and try to see if we can do some experiments to test whether yes or no TAT boundaries insulate every enhancer from every promoter, whether yes or no, if you change the contact probabilities between an enhancer and a promoter, you change the transcription levels and how you change them, and whether we could show any causality in the process. So I don't think we really were able yet to prove formal causality, but the evidence that we generate, I think it's pretty compelling. That and I refer specifically to one project that we did here, where we were able to show that if you change the contact probabilities between an enhancer and a promoter through changing their distance in the absence of any other confounding effects, you do change transcription levels substantially, and that there is actually a relatively smooth function that converts contact probabilities into changes in contact probabilities into changes of transcription levels, and this was I mean, probably one of my favorite projects ever. So it was always my dream project, I have to say. And Jessica Azuin in the lab, who joined, she was the first postdoc joining my lab, and she took up a super ambitious project, which eventually ended up well.
In nature, yeah.
Yes, exactly. But it wasn't for granted, for sure, yes. So because the idea there was actually to try to move an enhancer away from its cognate promoter and to insert it in many different positions in the genome in a place in the genome without any other confounding factors. So no CTCF looping, no other enhancers, no other promoters, but at the same time to do it in a place where there weren't any heterochromatic histone modifications, or not in a lab.
Which is not easy to find, probably.
No, there's not so many of such places. Not so many such places. So we found 2 or 3 in mouse embryonic stem cells, which is the system that we use. But they weren't good enough in the sense that there's still some residual CTCF sites here and there that actually make loops, even in boring places in the genome. So we had to also clean up this region from CTCF sites in order to have sort of a 500 or so kilobase playground where we could do this dream experiment. Something. And
the transcription was then purely dependent on the distance that the enhancer moved away from the promoter?
Yes, I think so. In this very very simplified locus, which actually doesn't exist endogenously, so it's a totally artificial system. But I think one of the things I learned in physics is that it's also good to try to simplify and reduce the number of degrees of freedom to the minimum if you want to isolate some effects. So at least in these conditions, if you change the distance between in this case, the SOX2 enhancer and SOX2 and SOX2 promoter, you do monotonically decrease. So if you increase the distance, you do monotonically decrease the effect that the enhancer has on the promoter, and the function that sort of so the correlation between changes in contact probabilities and changes in transcription levels is also very interesting, because it's actually a sigmoidal function that hence allows to some extent to buffer away changes in contact probabilities when the enhancer and promoter aren't too far away in linear genomic distance, but allows them to transmit smaller changes in contact probabilities to much larger changes in enhancer effect when they are further away. So typically now, at least I'm talking a little bit too causally, maybe. The correlation is not necessarily causation, but this is what we observed, one only change in the contact probability, essentially. So yes, and we think that this might be a very elegant way of making sure that an enhancer and promoter do talk to each other while they're separated by distances that are not too big, typically inside two TAD boundaries, but also to get insulated by TAD, but the small changes in quantum probability you get at TAD boundaries when they're far enough in the genome, in genomic space. And and then we have a few ideas of how this nonlinear relationship might occur. They have to be tested. So the one that we propose actually suggests that there might be some cooperative effects somewhere, very similar to also what Alistair Böttiger proposed in a almost companion paper on eLife, where they also performed some modeling of this effect. So we both think that there might be some sort of cooperative effects. So either chemical cooperativity or biochemical cooperativity, like in the case of Alistair, or some futile cycles that he also proposed. So in the way, so what we actually think might be happening, but this still needs to be tested, is that sequential interactions between an enhancer and promoter so a few of them might actually be needed in order for some processes downstream to actually happen with high efficiency and hence transmit the information to the promoter. And this might be what drives cooperativity in the system. But we have to test this, and notably using live cell imaging. I think this will be key. Yeah.
Yeah, then you investigated TAD function further and combined it with your earlier focus and looked at the TAD neighboring that of the non-coding transcript XIST, which controls X chromosome inactivation. So what did you exactly do to investigate this TAD function here, and what were the results then?
I presume you're referring to my postdoc work here, because I think so this is still the polymer simulations that I did in EDITH Lab. I think so. What we did, I mean, the connections between what I did back then and what we're doing right now is that now we know, back then when we used polymer simulations to deconvolve the single trajectory of the chromatin fiber at the XIST TSIX locus, we found that they are highly variable, as I said before, but we had no ways of sort of looking at their dynamics in living cells. So we always left with the question, how much time does it take for one cell to change its conformation of the chromatin fiber within a TAD from an elongated one to a compact one? So this was the question we had already several years ago, and there's no other way of knowing that except that they're actually measuring it using a live cell microscopy, which is actually what we ended up doing in my lab as well. And we were able to show that the conformation of a TAD, at least of the TAD that we're looking at, is actually highly variable in time and with context between contacts. I should say maybe physical proximity between sites that are separated by 150 kilobase in the absence of any intervening CTCF driven loops that only last for a couple of minutes and occur every 10 or so minutes. So this is also very much in line with what Anders Hansen has published last year. So it was sort of our two papers were out last year. We both measured the dynamics of chromosome looping inside the TAD, looking essentially at two viewpoints in live cells and monitoring how long they take to become close in space, how long it takes for them to separate again, and how this sort of fits with what we know about the dynamics of loop extrusion in living cells based on in vitro and in vivo measurements that were previously performed. So I think this is very important as well, because it sort of showed that. So it sort of it sort of shows that the single TAD is not just so again, it's not a blob. It's actually a very variable. So we already know it was the conformation of chromatin fiber inside TADs were very variable from cell to cell. But now we also know that it's also very variable in time. And if you look at a mouse embryonic stem cell over the 15 hours or so of its cell cycle, we know that interactions within a TAD between two loci that are separated by 150 kilobases or so, which is maybe the representative distance of enhancer promoter interactions as well, sort of assemble and disassemble so often that the time it takes for them to assemble and the time it takes to disassemble are infinitesimally shorter than the cell cycle, which also might suggest. So if I can something that also enhancer promoter interactions, although we haven't measured them yet, are also dynamic, and which adds a new interesting layer to the
Yeah, I just wanted to ask, right? There is at one point there is like in 3C and high C and all the experiments that you're doing that are on a population level, there is this average over all those cells, and now there is also like this second dimension or third dimension that is time, right? So
exactly.
you have to control for all of that.
Yeah, no, indeed. I mean, the time, of course, I mean, we appreciate more and more thanks to the work of many labs actually have used live cell microscopy in single cells and a single molecular resolution that the processes that occur on promoters and enhancers are dynamic and transient and highly stochastic. So promoter bursting is, of course, if you look at average level of expression of genes, this sort of hides population averaged and time averaged. Sorry, if you look at population averaged mRNA levels, this hides the time variability that occurs in single cells, where each promoter essentially bursts in time, alternating periods on and off. And we know that the processes that occur at the promoter are also stochastic. Transcription factor binding is very transient. Assembly of cofactors is also transient. And what we do not know is how all these processes are integrated in time in order to sort of give the desired transcription level, either in time or at the population level. And what we now think we are starting to see, although, I mean, we haven't measured enhancers and promoters directly yet, so I'm trying to do this, of course, is that it's also this dimension of enhancer promoter contacts or physical proximity that must be required to transmit physical biochemical information is also transient and dynamic. So it will be very exciting to see in the next couple of years how much dynamics there is in the proximity between an enhancer and a promoter, whether this relates to any extent with the onset of a burst or they're totally uncoupled and how.
You just mentioned the dynamics of promoter activity. What is this relative to? Is it only the cell cycle, or what other factors play a role in this that you maybe have to look out for in those experiments?
I think the main both Anders and our experiments show that what really controls the timing of interactions is the loop extrusion activity of cohesin. This is cohesin and CTCF. So if you degrade cohesin in mouse embryonic stem cells where we do our experiments, we see that the distances, but this was known, the distances between loci actually become larger. But we also see that their interactions now become way less frequent. At the same time, on the opposite side, on the contrary, if you have CTCF sites that face each other, flanking the viewpoints that we use for imaging, then we see that these viewpoints essentially lock on top of each other for much longer because of the formation of a stable loop between the two CTCF sites that is mediated by extrusive cohesin. So, and both Anders and our experiments agree on the fact that the duration of such a molecularly looped state is between maybe 10 and 30 minutes. So it's longer, but it's transient. And clearly the presence of CTCF sites and of course of extrusive cohesin makes these encounters very different. Yes. So it is.
No, go ahead.
No, that's more. Yes.
So what is it what you're working on currently, and what is your plan, let's say, for the next 5 years, or like the next grand application?
Well, let's see. So one of the things we are really interested in understanding is whether these findings with the SOX2 enhancer and promoter can be generalized to other enhancers and promoters, of course. So does any enhancer promoter pair, whether cognate in the genome or totally unrelated, show the same nonlinear relationship when you change contact probabilities? And does transcription change with such a strongly nonlinear function of the contact probability? So this is clearly one of the highest priorities for us, is actually to extend these experiments to more promoters and more enhancers, and if possible to also extend them technically by doing library type approaches in the genome. And this is one of the main limitations at the moment. It's very difficult to insert very large numbers of different sequences at different positions in the genome, because you always rely on relatively low insertion efficiencies, whether you use CRISPR or transposes. So the numbers are always in the low % or even less, actually, most of the times. So this is something we're really working on very much, and we hope to be able to to put up some new measurements soon somehow. And another thing we're really pushing hard on is live imaging of both chromosomal interactions and the molecular processes that occur at enhancers and promoters, notably transcription. So we would really like to measure whether enhancer promoter proximity correlates with bursts or not at single cell, single molecule level, and whether and how this is changed if you now change the frequency and duration of contacts between the enhancer and promoter, for example, by modulating either cohesin levels or introducing CTCF loops. So we really would like to sort of keep combining a little bit the single cell, single molecule view with more systematic unbiased measurements that use genome engineering to actually look at how, if you vary one or two variables, for example, the distance, or if you change the contact probabilities alone without changing the distance, if you change the sequence of the promoter, the sequence of the enhancer, the CTCF looping pattern, what happens to transcription? So the idea would actually be really, to combine these levels and keep using modeling, of course, to bridge between the two in order to reach maybe more quantitative understanding of long-range transcriptional regulation. Because the hope is that there are general rules. So it's not all about which sequence you're looking at, which enhancer you're looking at specifically, which promoter. Maybe there are, of course, but my hope is always that, as we saw in the case of SOX2 and enhancer and promoter system, and that if you sort of do unbiased measurements and in conditions where you can minimize confounding effects, you can find maybe quantitative general relationships that control long-range transcriptional regulation. Let's see if this is really true.
So to finish off this interview, I have two more general questions. The first one, did you at one point of your career face the situation that you have reached a dead end or did not know how to proceed to unravel the questions you wanted to answer?
Not yet, luckily not. I think it's very exciting. I think it's a very exciting ride since I started my PhD. I think so far it's been really, really fantastic. I think so. No dead ends. I also, because I think I'm lucky enough to work in a field that is very dynamic and where things happen every few weeks. So there's always new discovery, new concept, and so this really makes it very exciting. And also the ability to work in an environment that really sort of supports our interdisciplinary approach really makes it exciting. So when things don't go well in the lab, you always have some models that you can focus on and that bring you inside, and then you can think about new experiments to test those models. So it is really exciting, I think. And so dead ends, not really. Maybe when I started at the FMI, it was really a daunting task to set up a full experimental program because I'm, as I said, I'm a physicist by training. So of course I did quite some experiments during my PhD and postdoc, but I'm not a full experimental person. But I'd say, I was between the environment provided by the FMI and the people who were maybe crazy enough back then to actually join my lab. This worked fantastically well, and it's their merit, not mine, if the experiments have worked so well. So yes, no real dead ends, I would say. Okay.
So in the last 38 minutes, we have taken a journey through your scientific career. Can you maybe give a short summary about your most important findings or something that we might have missed in this interview?
Yes, I think, what can I say? Our most important finding, I think, my lab's most important finding, I think, is that there are maybe, at least we identified one general, I would say, I wouldn't call it law, but relationship that links two quantities in live cells. Specifically, this is transcription output and contact probability between an enhancer and promoter. And I think this is key because it sort of provides some sort of quantitative framework to explain how an enhancer might activate a promoter at a given distance. And also reveal that if you do experiments that are quantitative enough, you can get quantitative outputs that can be even interpreted using models. But it's hard to reduce this is the most important finding. I think the most important thing that we've done, I believe very humbly, is that we managed to combine both the single cell live information of processes that happen in single cell in time with what we think might be the mechanism that give rise to this general relationship. So I'm very proud of my lab for this. Yes.
Yeah. Thank you, Luca, for your time and for being on the show.
Thank you so much in the first place.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.